Hello friends, I am Shubhangi Rohatgi, PGT Biology, KV1 AFS Hindon, and I welcome you all to the first session of chapter 6 of class 11th biology, that is anatomy of flowering plants. Anatomy. We all know that anatomy is the study of internal structure of an organism. So, when we study anatomy, we study about the tissues, cells and various structural organizations of the body. Well, we know that the structure relates with the function. So, knowledge of anatomy is used for understanding various life processes. Also, the anatomical studies of plants throw light on their evolutionary relations as well as their environmental adaptations. Now, what happens is that generally students, pupils, they love to read and learn about the analytical biology vis-a-vis biochemistry and biotechnology and all, but when it comes to the study of morphology and anatomy, they seem to be afraid of them because the content is more descriptive and the people think that it needs to be crammed up. So, well, in today's class we will try to assign a reason to all the descriptions so that the need for the cramming up can be done away with. Let us begin. So, talking about the tissues, we all know that the tissues are group of cells having common origin and they usually perform common function. Plant tissues can be divided into two groups, meristematic tissues and permanent tissues. Let us take meristematic tissues first. The word has been derived from meristos, that is a Greek word, and it stands for divided. Growth in plants includes two stages. First, the production of new cells, and secondly, the expansion of these cells via uptake of water by the vacuoles. Now, what happens is that the cell division occurs only in the meristematic regions, while expansion may occur anywhere else. Let us see here in this diagram. We had referred to this diagram in earlier chapter also. So, here in this diagram you can clearly see there is a region of meristematic activity. It is the region where new cells are being continuously produced by mitotic division. The cells of this region are continuously dividing and chopping off new cells. These new cells are all alike, all very similar to each other because none of them are specialized. So, they just chop off a lot of new cells all through the time, and when these new cells enter the system, they first get elongated and later they get mature, that is they are assigned their specific function and structure accordingly. So, this is the region where they differentiate. We are talking about the meristematic tissue which is placed here or in other portions of the plant also, as we will see earlier, as we see later. So, coming back, the meristematic tissues are responsible for the division of new cells by mitosis and thus they are responsible for the growth of the plant. So, we all know that plants have indeterminate growth, that is the plants have the ability to keep on growing throughout their life, while the animals do not have this property, they have determinate growth. Can you think why the nature has given the property of indeterminate growth to plants only and not to the animals? Do you think that this property is to compensate loss of some other things that animals have but plants do not? Yes, we are talking about the mobility. Most of the times animals are in position to escape the dangers. They can simply move away and avoid the dangers, be it predation or natural disasters. On the other hand, plants are not mobile. They have to stand at one place. So, they cannot save themselves from herbivory or fire or any other natural disaster that is taking place. So, just imagine what would have happened if the plants had only determinate growth. Once a cow grazes by and eats up the shoots or the stems, the plant would have been dead. But it is not the case, thanks to the presence of the meristematic tissue. It is present there and it keeps on chopping off new cells for the growth of the plant, so that the plant body can continue to grow throughout its life. Moving on, let us now see the characteristics of meristematic tissues. The cells and the tissues we are talking about are formed exclusively by undifferentiated or embryonic cells. And these properties of the cells are as follows. They are relatively smaller in size. They are either rectangular or isodiametric, that is maybe polygonal or oval in shape. They are always compactly arranged and without any intercellular spaces. In fact, whenever you are asked to draw the portion of the plant or the cellular structure of the plant showing the meristematic tissue, you can draw them like these, because most of the time they are rectangular and they do not have any intercellular spaces. But what is to be kept in mind is this, that they have very large and conspicuous nuclei and dense cytoplasm. So, this is how we draw meristematic tissue. Wherever you are required to draw them like these. Yes, talking about the cell wall, their cell wall is thin and is made up of only cellulose. They have large amount of dense cytoplasm also. So, while you draw, please show some cytoplasm also in your diagrams, this way. Moving on, as I told you earlier also, that they have a single conspicuous nucleus situated in the center of the cell. The vacuoles are either absent, or if they are present, they are very small in size, because the cytoplasm is dense. So, the vacuoles are either absent, or if present, they are very small. And no ergastic substances are also present. That is, ergastic substances are non-protoplasmic substances like reserve food materials or secretory products or excretory products. They are absent from these meristematic cells. Finally, most of the organelles are also absent except for the mitochondria. Mitochondria is of course there, because it is there to provide the energy for rapidly dividing cell. But other organelles are either absent or present in non-functional state. For example, plastids. Plastids are present in non-functional state called proplastids. This was about the characteristics of meristematic tissues. Let us now study the different types of meristems that are present. Actually, the meristematic tissue is present all through the plant body at different regions. So, there are many ways in which we can group or classify the meristematic tissue. The meristematic tissue can be classified on the basis of their origin, location in the plant body and differentiation also. So, let us discuss the first type of classification, that is based on the origin. When we talk about origin, the meristematic tissues can be divided into promeristems, primary meristems and secondary meristems. Promeristems are the ones which are present in germinating embryos or germinating seedlings when they are very young, and the derivatives of these meristems, they provide new cells for the plant bodies. Some of these cells give rise to the primary meristem also. The primary meristem in turn divides to form the primary plant body, the young plant body, and some of the cells of the primary meristem give rise to secondary meristem during later periods of growth. The secondary meristem is also very important, because it causes the secondary growth, the wood formation. Have you ever noticed this thing, that whatever be the age of the tree, whatever may be the girth of the tree, however woody it may be, but when it is a young plant growing out of the soil, when it is just a young sapling or even a few weeks older, it is herbaceous. All the plants, all the trees, they are herbaceous when they are young. There is no wood formation. The wood formation takes place only after a certain period of growth has been attained. So, the period before the wood formation, it can be called as the period of primary growth. Here the primary body is forming, the organs are forming, the elongation is taking place. On the other hand, wood formation is secondary growth. Here the increase in girth of the plant takes place. Here the wood formation takes place. So, the secondary growth is associated with the secondary meristems. Okay. So, primary growth is associated with primary meristems and secondary growth is associated with the secondary meristems. So, this was what I was talking about earlier, primary versus secondary growth. As I had just said that primary growth is because of activity of primary meristematic tissue, while this includes the activity of only secondary meristematic tissue or by joint activity of both primary and secondary as well. It increases the height of the plant, whereby secondary growth increases the girth of the plant. So, it results in radial growth. It is the first growth of the plant and its parts, while it is a later period of growth. The primary growth stops after complete tissue differentiation has taken place. On the other hand, secondary growth occurs only after the part of the organ has completely developed. Primary growth occurs in all plants and in all parts, but secondary growth is not seen in monocots. This is the diagram of apical meristems that is given in your NCERT. We will come to it later when we will discuss the types of meristem on the basis of position. On the basis of position, the meristems can be divided into apical, intercalary and lateral. Let us see in the diagram here. This diagram is in the class 9 textbook of NCERT. Here you can see the apical meristem, which is present at the apices. It can be present at the shoot apex or the root apex also. Then the intercalary meristem. The intercalary meristem is present at the nodes, and the lateral meristem runs parallel to the plant body and it is somewhere in the lower regions of the plant. Moving back. So, the apical meristem, as I said, occurs at the apex of roots, stems and leaves. It constitutes the actively growing regions of the plants and results in growth of height of the plant. It includes the promeristem and the primary meristem. The intercalary meristem, this is special because this is placed between two permanent tissues. Actually, what happens is that it must have been the part of initial meristem, but because of growth of the other tissues and cells, it has been separated and now it is known as the intercalary meristem. When we draw the meristems, we draw them like these. So, here I have shown the apex. I can draw the apical meristem here. This can be the shoot apical meristem, SAM. Then there are the nodes, and from nodes arise the leaves or branches, and intercalary meristem is present at the bases of these nodes or even in the leaf sheaths, but only in monocots. So, this will be intercalary meristem. These two types of meristems, they divide to produce increase in height of the plant body, while there is another meristem that develops later in the life of the plant, and it is in parallel to the long axis of the plant body. When it will divide, it will produce cells this way and this way. What will happen? The girth of the plant will increase. This meristem, which develops later and causes increase in the girth, is known as lateral meristem. So, these are the three types of meristems based on the position in the plant body. Let us move on to the permanent tissues now. What are permanent tissues? Here the cells are structurally and functionally stabilized and specialized also. That is, the cells which were chopped off from meristems have grown and matured and have acquired their particular function and structure accordingly. That is, they have been differentiated. So, permanent tissues are those tissues whose cells are differentiated and they also have lost the ability to divide. The permanent tissues in the plant can be simple or complex depending upon the types of cells they are made up of. If the tissue is made up of only one type of cells, then it is known as simple permanent tissue. On the other hand, if it is made up of more than one type of cells, then it is known as complex permanent tissues. You have studied about some permanent tissues in your lower classes also. So, the simple permanent tissues can be parenchyma, collenchyma or sclerenchyma. Sclerenchymatous tissues can also be in form of fibers or sclerids. We will take them up one by one. Beginning with the parenchyma. This image is there in your class textbook. The parenchymatous cells. Look at the shape of the cells and note the presence of intercellular spaces. So, the cells are isodiametric and the cell walls are quite thin and they are made up of cellulose, and they may have some intercellular spaces present amongst themselves. This is a microscopic image of parenchymatous cells, and look here, you can clearly see the intercellular spaces and the cell walls. Please note them, how thin they are. Let us talk a little bit more about the parenchyma. Para means beside and chyma means filling. So, this is the cell which actually makes up the whole plant body in form of fillers. It fills up the plant body. So, it is present everywhere, be it the cortex and pith of the stem, cortex of the root, mesophyll of the leaves, pulp of the root, and even the endosperm of the seeds. The cells here are living and they are capable of division if stimulated. In normal conditions, they won't divide, they have grown and acquired their function, they are differentiated, but if stimulated properly, they can divide. For example, if there is any wound, so they will divide to repair the wound. They have thin but flexible cellulose cell walls and they are generally polyhedral when closely packed, but can be roughly spherical when isolated from their neighbors. And also the vacuoles are very large and these vacuoles are used to store and regulate ions, waste products and water. Let us now study the functions of the parenchyma. Since they are present everywhere, they perform a large variety of functions. For example, when they are present in the leaves and they bear chloroplasts, then they are known as Sclerenchyma, and then they take part in various activities of photosynthesis. Then most of the parenchymatous cells are specialized for storage of food. They can store starch, proteins, fats, oils and even water in roots, seeds or in cotyledons also. Some of the parenchymatous cells are specialized for secretion, as in parenchymatous cells lining the insides of raisin ducts, As I said just now, that some of them are also involved in wound repairing, and some others have specialized functions like aeration. These parenchyma cells, they are present in the aquatic plants, and there they provide the buoyancy. Here is a picture showing the parenchyma, and the parenchyma not only serves the purpose of gaseous exchange, but as I just said that in aquatic plants it also serves to maintain the buoyancy. These are the finger like cells of palisade parenchyma. These dots represent the chloroplast, so they take part in photosynthesis and known as Sclerenchyma. These are spongy parenchyma cells. Here you can see large air chambers, so they help in gaseous exchange, and since the chloroplast is also present, so they take part in photosynthesis also. Moving on, as I had said that this forms the filler tissue. So, here is a TS of the stem, and you can see the central part of the stem, that is known as the pith. It is largely made up of parenchymatous cells. Then some parenchymatous cells are also specialized for storage. Here you can see the starch grains being stored in the parenchyma. Moving on, let us see the Collenchyma. The Collenchyma is present below epidermis in dicots, and there are thickenings at the corner. These thickenings are made up of cellulose, hemicellulose and pectin. They may have sometimes chloroplast also. Intercellular spaces, however, are absent, and they provide mechanical support. Here in this microscopic image, you can see the hypodermis, and here in the cells of hypodermis at the corners, can you see the thickenings? See here, these are the thickenings that are present in the Collenchyma. So, the cells here are elongated and the cell walls are irregularly thick. Their main function is to provide structural support, especially to the growing shoots, because when the plant organ is growing up, it needs mechanical support, but it needs elasticity as well, because it has to extend. The cell walls cannot be very stiff in these regions. Why? Because otherwise the growth will be hampered. So, Collenchyma is present in the regions where the growth is taking place, young parts are being formed, and the Collenchyma is there to provide mechanical support as well as the elasticity. So, the cells here are usually living because the cell walls are not very stiff, and the cell wall thickness is strongly affected by mechanical stress. If the plants are under stress, the mechanical stress, suppose the wind is blowing very hard at a place, then at those places or under such conditions, the Collenchymatous walls will be 40 to 100% thicker than those plants where there is no such stress. Also, Collenchyma cells can be very conspicuous under microscope because they have high refractive index because of their thickened cell walls. The next permanent tissue is Sclerenchyma, and these tissues have dead cells and they do not have protoplast. The cell walls are thick and they are highly lignified. Even they may have pits also. What is their main function? They provide mechanical support. This image is there in NCERT. You can see Sclerenchymatous cells are of two types, fiber or sclerids. So, this is a fiber and this is a sclerid. And here in the TS of a stem, you can see the Sclerenchyma. Please note the thick walls. Actually, what happens is, suppose this is a cell and now the thickenings are taking place. So, the cell wall has become lignified and a lot of lignin has accumulated in the walls. What will happen? the cell walls will become thick while the lumen will become narrow. This is the lumen. This is how a sclerid looks. Same is the case with the fibers also, with thick walls and a very narrow lumen. To differentiate between sclerids and fibers, sclerids are broad, have blunt end walls, they may be branched, and they are formed by secondary thickenings of parenchyma. Just what I drew here, that parenchymatous cells, they develop thickenings, and because of these thickenings the lumen became very narrow, and now the cell is known as a sclerid. On the other hand, fibers are elongated and they have tapering ends. If I have to draw a fiber, I will draw it like this. So, these are the tapering ends, and they are unbranched and they originate from the meristematic tissue. This was all about the simple permanent tissues. Moving on to the complex permanent tissues. Complex permanent tissues, I shall recall, are those which have more than one type of cells, and here we will study about xylem and phloem. The xylem elements, as I just said, that complex tissues have more than one type of cells, and the cells here in xylem are known as xylem elements. The word xylem has been derived from a Greek word xylos, which stands for wood. As we know that xylem is mainly responsible for conduction of water and mineral salts from roots to other parts of the plants, and xylem which is formed earlier from the procambium is known as primary xylem, while the one which is derived later from the vascular cambium is known as secondary xylem. The four elements of xylem are tracheids, vessels, xylem parenchyma and xylem fibers. We will study about them one by one. This is a figure of tracheids given in your NCERT book also. What are tracheids? Tracheids are imperforate cells with bordered pits on their end walls, and they are arranged one above the other. Look here, when we draw tracheids, tracheids are just like fibers also, but their lumen is much broader. So, we can draw a tracheid like this. This is one tracheid, and how is it arranged in the plant? It will be arranged with the other tracheid. This is how tracheids are arranged inside the plants, with their tapering ends and their bordered pits. These pits help in flow of water from one tracheid to another. So, conduction of water and mineral salts takes place through the bordered pits. Please note here that their end walls are closed. So, the water or the salts have to move from one tracheid to another, and so on and so forth. As I said that they are just like fibers, but their lumen is broader than that of fibers. The secondary wall is lignified. So, they also offer mechanical support. Wherever there is lignin, remember there will be support, because lignin forms quite a hard covering on the cell walls. The cell wall thickenings can be of different types. They can be annular or scleriform, and tracheids are the chief water conducting elements in gymnosperms and pteridophytes. They are also present in the angiosperms, but in gymnosperms and pteridophytes they are the chief conducting elements. The vessels are not present there in gymnosperms and pteridophytes. Moving on to the next element, that is the xylem vessels. This is a diagram of xylem vessel, and here also you can see there are so many pits, and the pits are present on the cell wall end also. So, the lumen is much wider than that of tracheids. Vessels are chief conducting elements in angiosperms. Tracheids are also there in angiosperms, but vessels are the chief conducting elements there, and they are absent in pteridophytes and gymnosperms. They offer mechanical support also, and they occur parallel to the long axis of the plant body, forming up a tube. You can say that we draw a vessel like this. Suppose this is a vessel. So, vessels are arranged end to end, one above the other, making long parallel tubes inside the plant body. The perforated plates at the end wall separate the vessels, and these perforated plates may be simple or multiple with many pores. This is the perforation plate. The other two elements of the xylem are xylem fibers and xylem parenchyma. This is a picture of xylem parenchyma and a xylem fiber. Xylem parenchyma stores food and tannins, and it also allows radial conduction of water. The fibers of Sclerenchyma associated with xylem are known as xylem fibers. They give additional mechanical support to the plant body. They are present both in primary and secondary xylem, and xylem fibers are dead cells and have lignified walls with narrow lumen. So, here if I ask you to differentiate between a fiber and a tracheid, what will be the points of difference that you will mention? We have discussed just now that both have tapering ends, both have lignification on their cell walls, and both may have pits also. So, what is the difference between the two? And yes, both of them of course provide mechanical strength also to the plant. What is the difference? Difference is that the tracheids have a wider lumen as compared to the fibers. Fibers are quite narrower when we compare them to the tracheids, and tracheids perform the function of conduction of water in xylem, while fibers they only provide mechanical support or mechanical strength. What about the xylem parenchyma? Xylem parenchyma are normal parenchymatous cells, and they are present between the xylem elements. They form medullary rays also, and they help in radial conduction of water. Radial conduction means, look, suppose this is a plant, the water needs to go up, this we all know, from roots to other plants of the parts of the plant, but since the xylem is here somewhere in the middle, the water also needs to be taken in other directions towards the periphery, and this is the radial conduction of water, and this is done by xylem parenchyma. Xylem, the primary xylem, it can be of two types, protoxylem and metaxylem. The protoxylem is the one which is formed by primary xylem, and it develops before the plant organ has completed its growth. Suppose we are talking about xylem of the stem or xylem of the leaf. So, protoxylem will be formed when the growth is not complete, this growth is still going on, the stem is still growing or the leaf is still growing. At that period of time, it will have protoxylem elements. So, the protoxylem is comparatively less prominent. It is made up of smaller and narrower elements. What does that mean? That when we see this protoxylem elements in a transverse section, we see that the cavities made by protoxylem elements are comparatively smaller. That is what I mean when I say that the protoxylem elements are inconspicuous or comparatively less prominent. They are made up of smaller and narrower elements. So, this is protoxylem elements. The wall thickness of the conducting elements are of simple and primitive type, that is annular or spiral. Annular thicknings or spiral thicknings, suppose this is the element, and if some thicknings like that are present, then we say that these are the spiral thicknings. So, protoxylem elements have spiral thicknings on them and they are capable of being stretched. Obviously, they are present in the growing organs. The organs are growing, they are expanding. So, the protoxylem also needs to expand with them. That is why the protoxylem elements are capable of being stretched. That is, they can undergo growth. Here in protoxylem, the fibers are absent or rare. So, can you tell me in this microscopic image where are the protoxylem elements? Yes, the arrows show them. These smaller elements are the protoxylem elements, while the xylem, which develops later and has larger elements, is the metaxylem. So, if we draw comparison, we can say that it is the later formed xylem. It differentiates when the plant organ has completed its growth. Now, once the growth has been completed, the size which was to be attained has been achieved. Now, the protoxylem will stop developing, but metaxylem will start to differentiate in that fully grown mature plant organ. Metaxylem is quite prominent. It is formed of broader and larger elements. Here you can see, notice the difference between the size of the elements in metaxylem and protoxylem, which are too small, while the metaxylem elements, they are so big. The wall thicknings also are very complex and they are of advanced type. They are reticulate, scleriform or pitted. Metaxylem elements cannot be stretched. Obviously, they have been formed after the growth has been completed. So, they cannot be stretched much. Fibers often occur in metaxylem because this is fully formed xylem and it provides mechanical strength also. So, fibers are also there. Now, talking about the exarch or endarch condition. Have you heard these terms before? Endarch xylem is one where the protoxylem elements are towards the pith. Here in this stem section, you can see that the metaxylem is towards the periphery. The larger elements, please, to an unknowing eye, these will seem just like holes in a diagram. But I want you to remember how xylem actually looks like under the microscope. That is why instead of taking the diagrams or photographs, I am using the microscopic images. So, whenever you look under a microscope while cutting the sections of stems and roots in your laboratory classes, you should be able to identify which cell is that of xylem and which one is that of phloem. So, here please notice that the ones which larger cavities, these are the metaxylem elements, while these towards the inside are the protoxylem elements. And such an arrangement where metaxylem is towards the periphery and protoxylem is towards the pith is known as endarch condition. It is present in the stems, while on the other hand, the exarch condition is opposite of it. Here the protoxylem is towards the periphery while the metaxylem is towards the center. Such condition is known as exarch condition and it is present in the roots. So, you will differentiate and remember the difference between endarch and exarch condition on the basis of protoxylem only. When the protoxylem is towards the center, it is endarch. End means inside. So, protoxylem is towards inside, while eggs for outside, remember it like that. So, protoxylem is towards outside. It is present in the roots. This was all about the xylem elements. Let us take now the phloem elements. In trees, the phloem is the innermost layer of the bark, hence the name. It is derived from the Greek word phloios, meaning the bark. What does the phloem do, by the way? Yes, you are right. The phloem carries the photosynthetes to different parts of the body of the plant. The elements of phloem originate from the procambium of apical meristem or even from vascular cambium, and as was the case with xylem, same is with phloem also. When it originates with the apical meristem, it will be primary phloem. When it originates from the vascular cambium, it will be secondary phloem. Phloem is also a heterogeneous tissue, that is a complex tissue, that is what we are discussing. It is made up of 4 different types of cellular elements, namely sieve tubes, companion cells, phloem parenchyma and phloem fibers. Let us take these one by one. Sieve tubes and companion cells are directly involved in translocation of organic substances, while these provide stability, structural maintenance and conduction of other things. Sieve elements. Now, sieve elements are of 2 types, sieve tube cells and sieve cells. Sieve elements are the conducting elements of phloem. They have thick primary walls and they are living cells with an intact osmotically active plasma lemma, some mitochondria and endoplasmic reticulum. So, remember in xylem the tracheids and vessels, they were dead cells and tissues, they had just an empty lumen. On the other hand, when we compare phloem with that, the phloem cells, they are living, the conducting cells, they are living and they have plasma lemma, mitochondria and endoplasmic reticulum also. However, when they are in conducting state, they lack cytosol, ribosomes, Golgi apparatus, nucleus, even the central vacuole. So, the lumen is mostly free for the phloem sap movement. A special protein named P protein is also present in the phloem and it is formed in the sieve elements of many plants. It contains lining layer of cytoplasm and it is very important feature of sieve elements because conduction of food materials takes place through these cytoplasmic strands. So, just I had said that sieve elements are of 2 types, sieve cells and sieve tubes. Let us talk about sieve cells. They occur in pteridophytes and gymnosperms only. They have sieve areas on their lateral walls only and are not arranged one above the other in linear rows. Sieve cells, which may be compared to tracheids, are narrow elongated cells without conspicuous sieve areas and they are more primitive than the sieve cells. As I had shown you the arrangement of tracheids, this is how even the sieve cells are arranged. They are not arranged in a stacking order one over the other, but they are arranged sideways in the plants and they are more primitive. Albuminous cells, they are associated with the sieve cells and they have a similar role to companion cells. They develop from the marginal cells of phloem rays and are structurally and functionally associated to the sieve cells. How structurally? Through plasmodesmata, and how functionally? Because they help the sieve cells to conduct the phloem sap. Now, coming over to the sieve tubes. Sieve tubes occur in angiosperms. They are arranged one above the other in linear rows, just like the vessels, stacked over one over the other, and they have sieve plates on their cell wall. Please refer to the diagram here. You can see this is one tube cell and this is another tube cell and there is a sieve plate in between on the end wall, and sieve plate is named so because of presence of numerous pores. So, it gives them an appearance of that of a sieve, and they are associated with the companion cell. What is happening here? Actually, they have to conduct the phloem sap, and we know that phloem sap conduction requires energy, that is the ATP is required for loading and unloading of phloem. So, that means the conduction cannot take place on its own in the dead cells. That is why the sieve tubes are living cells, but they need lumen for conduction. So, the functions of the nucleus or cytoplasm of the sieve tube cell has been taken over by the companion cell. The companion cell works to keep the sieve tube cells alive. So, talking about the companion cells, each sieve tube element is accompanied by one or more usually dense cytoplasmic cells called companion cells. So, if this is the sieve tube cell, you can see the sieve plate here. So, this will be the companion cell. It has dense cytoplasm and a conspicuous nucleus. The companion cells lie along the sieve element and in fact are product of longitudinal division from a common mother cell. That is, the sieve tube cell and the companion cell both originate from a common mother cell. They play important role in loading and unloading of phloem, and also they provide ribosome-less sieve elements with essential proteins and ATP. They possess specific intercellular communications in their common walls and have several plasmodesmata. So, they are in continuous communication with the sieve tube cells with the help of several plasmodesmata that are present between the companion cell and the sieve tube cell. In gymnosperms, typical companion cells are lacking in the phloem, but then there are no sieve tube cells also in the gymnosperms. Instead, the sieve cells are present and the function of companion cell there is performed by the albuminous cells. So, sieve cells and albuminous cells are present in the gymnosperms, while sieve tube cells and companion cells are present in the angiosperms. These ones, the sieve tube cells and companion cells, they are more evolved as compared to the albuminous cell and sieve cell. Moving on, the phloem parenchyma. The parenchyma cells associated with the phloem are called phloem parenchyma. They are living cells and the cell wall is primary, composed of cellulose, just like the normal parenchymatous cells that we studied about in the earlier part of the chapter. They store starch and fats, and they also contain resins and tannins in some plants. They are present in all pteridophytes, gymnosperms and dicots, but they are absent in monocots. There are two types of parenchyma in secondary phloem, axial parenchyma and ray parenchyma. Axial parenchyma will be somewhat parallel to the periphery of the plant, while ray parenchyma will be moving from periphery to the center. Last element of the phloem is the phloem fiber. The fibers of Sclerenchyma associated with phloem are called as phloem fibers or bast fibers. Again, I repeat that these fibers are made up of Sclerenchyma, that is, these are the same Sclerenchymatous fibers that you studied about under simple permanent tissues. But when they are associated with phloem, then they are known as phloem fibers or bast fibers. They are narrow, vertically elongated cells with very thick walls and a small lumen, that is the cell cavity. Among the four kinds of phloem elements, phloem fibers are only dead tissue. So, the four elements of phloem are the sieve elements, then the companion cell, the phloem parenchyma and the phloem fiber. These four, out of these four, three are living, the sieve tube elements, the companion cell and the phloem parenchyma. These three are living. Please remember this, even though sieve tube elements and companion cells are associated with conduction of the sap, yet they are living. Only phloem fibers are dead. This is in stark comparison with the xylem, where most of the elements are dead. Trachees are dead, vessels are dead, even fibers are dead. Their only living element is the xylem parenchyma. And these phloem fibers are very important also. For the plant, they are for strengthening and supporting, and for us, they hold a commercial importance because these fibers are abundantly used for manufacture of ropes and cords. Can you give me some examples? You have studied in lower classes also about jute, hemp, flax. All these are phloem fibers. Protophlloem and metaphloem. Protophlloem is the one which is formed first and thus it has smaller sieve tubes. Metaphloem on the other hand is formed later and thus has larger sieve tubes. It is just like that of xylem and protoxylem and metaxylem. So, proto formed first and will have smaller cavities, be it xylem or phloem. Meta formed later will have larger cavities, both in metaphloem and metaxylem also. This was all about today's class. So, today we studied about the different types of tissues that are present in the plant body. We began with the meristematic tissue and we continued with the permanent tissue. There are a few questions I would like you to find answers for. First, does the classification of meristems on the basis of origin and position seem to overlap? How? The meaning of this question is that you have studied two types of classification, one on the basis of origin, other on the basis of position. Do you think that there are points, there are terms which are overlapping in these classifications? Please explain. The second question is, have you heard that meristem cultures are done in order to obtain virus-free plants? What characteristics of meristematic cells do you think help them to stay virus-free? It is general knowledge that viruses fail to grow and replicate in meristematic tissues. Can you think of the reasons why viruses fail to grow in the meristematic cells? Third, what is totipotency? Find about it. Which type of cells should be most suitable for tissue culture and why? Lastly, I would like you to differentiate between xylem and phloem. We have discussed a lot of points differentiating between xylem and phloem, and next Collenchyma and Sclerenchyma. Both types of cells have thickenings on the cell wall somewhere or the other. So, they seem to be similar, but I know you will be able to find out differences between Collenchyma and Sclerenchyma. In the next class, we will study about the various tissue systems, the systems of tissues. So, up till now we were just studying about xylem and phloem and parenchyma, collenchyma and sclerenchyma, but now in the next session we will study about the tissue systems, the epidermal tissue system, the vascular tissue system, and also the anatomy of dicot and monocot, stem, root and leaf, how the cells and tissues are organized in these parts of the plants. This was all about today's session. Thank you.